Greetings. My name is Sydney Y.K. Brown. I'm a graduating neuroscience student at Agnes Scott College, and I had the opportunity to conduct research at Meharry Medical College under the mentorship of Dr. Aramandla Ramesh and Dr. Kenneth J. Harris. Today, I will be presenting research I conducted while at Meharry on the expression of benzoate pyrene biotransformation enzymes in the spleen of a rat model of colon cancer. To provide a brief introduction, polycyclic aromatic hydrocarbons, PAHs, are a family of pervasive environmental toxicants produced by incomplete combustion and the breakdown of organic matter. Benzoate pyrene, BAP, is an example of a PAH compound and can be found in grilled red meats, tobacco smoke, and fossil fuel emissions. Drug metabolizing enzymes, DME, metabolize BAP into benzoate pyrene diol epoxide, a carcinogenic compound that results in DNA mutations. These DMEs can be placed into two categories: phase 1, which works to activate BAP, and phase 2, which facilitates the excretion of BAP through conjugation. Literature suggests that BAP metabolites can affect the amount of white blood cells, allowing immunosuppression to ensue as the membrane integrity of macrophages are compromised when mutations in genetic material arise. A vital component of the immune system and its largest organ is the spleen. The spleen filters blood and foreign molecules. Old red blood cells are recycled while lymphocytes and macrophages are stored to fight off infection. As BAP has immunosuppressive effects when metabolites are not excreted by phase 2 enzymes and have the ability to jeopardize the integrity of organ function in a biological system, our goal was to assess the expression of DMEs in the spleen of rats modeling colon cancer to observe the possible immunosuppressive activity of BAP present. To answer this question, we used a 7-week-old male and female polypoesis in rat colon rat model. These rats have a mutation on their APC gene causing the development of colon cancer and are known as PERK rats. Our experimental group was treated with 50 microgram per kilogram body weight of BAP for 60 days. After 60 days, the animals were euthanized and tissue and organ samples were collected. The spleen samples gathered were then homogenized and centrifuged individually. A Bradford assay was then performed to measure the protein concentration of each sample. Working samples were then subjected to gel electrophoresis and Western blot analysis, probing for phase 1 DMEs, cytochrome P450-1A1, CYP1A1, and CYP1B1. Phase 2 enzymes of interest were glutathione S transferases, GST, and sulfur transferases, SALT. Our major significant research findings were gathered from Figure 1 on the slide, which reflects the expression of CYP1A1 in control and experimental groups between sexes. On the top of the graph is an image from Western blot analysis of CYP1A1 in comparison to our housekeeping gene, GAPDH. The figure illustrates that control male PERK rats have an increased expression of CYP1A1 when compared to control female PERK rats, and female PERK rats exposed to BAP have a significantly increased expression of CYP1A1 when compared to male PERK rats exposed to BAP. Data gathered from Figure 1 suggests that unaltered cytochrome P450-1A1 expression in male PERK rat spleens likely dissuades benzoate pyrene-induced immunosuppression. Future directions would include repeating the BAP biotransformation enzyme expression assays with an increased number of samples. Further studies would include the use of HPLC to separate and quantify the BAP metabolites in each spleen sample while utilizing flow cytometry to detect and measure physical and chemical characteristics of a population of B lymphocytes as well as T lymphocytes.